This is one of the eye models we have in lab. I want to go over some of the major parts. This is a sagittal view, so you really don't see everything. But you can see the three tunics or layers, the fibrous outer layer, which is the sclera, and in the front it makes the cornea. You have the middle vascular layer, which you can see the choroid back here is pink. In the front it becomes a ciliary body, which connects to the via the suspensory ligaments to the lens. Over here you have the ora serrata, and in the front, when you remove the lens, you'll be able to see the iris. That's also part of the middle vascular layer. The hole in the iris, that's the pupil. Okay, I'll put this back. All right, so with the retina, of course, is the yellow. That's the innermost layer. That's the neural layer. That's where you're going to have the photoreceptors, the bipolar cells, the ganglionic cells. There's a couple areas of interest. You can see this tiny little depression here. That's representing the macula lutea, and the central pit of that is called the fovea centralis. You'll be learning in lecture that that's where you have a high concentration of cones, picking up high resolution imaging. Then you have this area of interest. This is called the optic disc, also known as the blind spot. So if light hits you in this particular area of the eye, you will not see it because there's no photoreceptors here. Instead, that's where all the nerves leave. When you turn this model around, you can see the cornea. You can even see the superior and inferior rectus muscles. Of course, there's other models that show you a better view of that, but those should be the major parts of this model.